Welcome back to Biology 2210, AMP1 at CNM. We are discussing Chapter 12, the nervous system, and we've made it up to objective number 136. And so we're in the midst of discussing synapses, and we've specifically just completed our discussion of types of neurotransmitters. And we finished off with the concept that neurotransmitters can be excitatory or inhibitory, and that's mostly based on the type of receptor that exists in the postsynaptic cell. And so even the same neurotransmitter can have both excitatory and inhibitory effects, again depending on its receptor. So there are a variety of different kinds of synapses out there with different kinds of receptors acting in different ways in response to binding to the neurotransmitter. As a small subset, just to give you a sense of the variety of types of synapses that exist, we're going to talk about three very common types: the cholinergic synapse, the GABAergic synapse, and the excitatory adrenergic synapse. And so that's where we pick up today at objective 137, discussing the cholinergic synapse, and specifically an excitatory cholinergic synapse. So a cholinergic synapse, by definition, is one that uses acetylcholine as the neurotransmitter. That's where the choline part of cholinergic comes from. And the excitatory cholinergic synapse is essentially a synapse like the neuromuscular junction that we looked at in our muscle chapter. So a lot of the elements of the cholinergic synapse should be quite familiar to us. So as we know, a action potential, a nerve signal arrives at the axon terminal, and instead of sodium flowing in through voltage-gated sodium channels, we know at the axon terminal it's calcium ions that flow in and cause the depolarization in the axon terminal. The calcium, as I mentioned back in the muscle chapter, enters the axon terminal and activates kinase enzymes which alter the fluid nature of the membrane, making it more fluid. This increase in fluidity is what triggers exocytosis of the synaptic vesicles, which, remember, are docked just beneath the cell membrane, and acetylcholine is released. As we know, acetylcholine diffuses across the synaptic cleft and binds to the acetylcholine receptors in the postsynaptic membrane. This can be, again, in the muscle cell, in the motor end plate, or if we're talking neuron-neuron type synapse, then it could be acetylcholine receptors on the dendrites, say, for instance, of the postsynaptic neuron. The binding of acetylcholine to the acetylcholine receptors changes their shape, and as it turns out, these receptors are also ion channels, specifically chemically gated ion channels that open in response to binding to this acetylcholine, and it allows the simultaneous flow specifically of sodium and potassium. Because the gradient for sodium is greater than is the gradient for potassium, more sodium flows in than potassium flows out, causing a depolarization of the membrane. This depolarization of the membrane can occur in different sizes depending on how many and how long the gated channels stay open. So this creates a graded potential in the postsynaptic cell, which we call a depolarizing or excitatory postsynaptic potential. The sodium ions that are injected will spread through the postsynaptic cell, of course getting weaker in terms of their strength as they spread and become more dilute as they spread. But as we know, if the depolarization, if the excitatory postsynaptic potential is strong enough, by the time it reaches the trigger zone of the postsynaptic neuron, it will trigger an action potential in the next neuron. Of course, if this is a muscle cell, the postsynaptic potential, which is, remember, called a motor end plate potential, doesn't have to spread very far, because just outside of the motor end plate you have normal sarcolemma, normal muscle cell membrane, which contains voltage-regulated gates. And so it's very easy to get an action potential from a motor end plate potential in a muscle cell. But in a postsynaptic neuron, the depolarization has to spread, remember, through the dendrites, through the cell body, and it has to reach the trigger zone and still be strong enough at that point to cause threshold levels of depolarization. So this is all very familiar and essentially what we've been talking about for the last two chapters. And here we see a familiar diagram of the excitatory cholinergic synapse. Remember also, just one added detail, we do have acetylcholinesterase involved, and acetylcholinesterase helps to break down acetylcholine so it doesn't perpetually stimulate the postsynaptic membrane. Now, not all synapses work this way, and this is why we're talking about another couple of synapses, just to give you, again, a feel for how different neurotransmitters might work. We have another type of synapse called a GABAergic synapse, which employs gamma-aminobutyric acid as the neurotransmitter, and that's, of course, what GABA stands for, is gamma-aminobutyric acid. And so in this kind of synapse, again, we have a similar beginning to the synapse. The nerve signal triggers the entry of calcium into the axon terminal. The calcium stimulates enzymes or activates enzymes which activate exocytosis, and the synaptic vesicles containing GABA will release GABA into the synaptic cleft. GABA then will bind to its specific receptor in the postsynaptic membrane. In this case, though, the receptor is not a chemically gated channel that opens and allows sodium and potassium to flow, but instead, for GABA, its receptor is a chemically gated ion channel that is specific to chloride ions. Remember, chloride ions are negatively charged ions. And when we open the chemically gated chloride channels by GABA binding to them, chloride will move across the membrane. Now, the concentration gradient of chloride ions is that chloride is greater outside the cell than inside. This is largely because chloride follows sodium, the positive charge of sodium as it's pumped out of the cell. And so opening the chloride channels causes chloride ions to enter the cell along its concentration gradient. And of course, the entry of negative charge makes the inside of the membrane more negative than it was when it was at rest. This kind of change, which makes the membrane more negative than it was at rest, remember, is called a hyperpolarization. So the binding of GABA ultimately causes a hyperpolarization. Well, of course, this takes the membrane potential farther away from threshold. It's going in the opposite direction of depolarization. And if I make the postsynaptic membrane more negative, more hyperpolarized, in other words, take it farther away from threshold, then I make it more difficult for that postsynaptic membrane to reach threshold and be triggered. And so this ultimately has an inhibitory effect on the postsynaptic neuron, and it becomes less likely to fire. So this is what we call an inhibitory GABAergic synapse, which has an inhibitory effect. And so again, we see that neurotransmitters can have excitatory effects by causing depolarization of the postsynaptic membrane, or they can have inhibitory effects by causing hyperpolarization. An even more complex kind of synapse is the adrenergic synapse. The adrenergic synapse is the synapse that is going to involve norepinephrine or noradrenaline as the neurotransmitter. Remember, an older name for epinephrine is adrenaline, and an older name for norepinephrine is noradrenaline. And that's where the name adrenergic comes from the adrenaline, original adrenaline name. And so the adrenergic synapse, which involves norepinephrine and any of the other monoamine and neuropeptide neurotransmitters, act through what are called second messenger systems. Now, I don't know that you've been exposed to the concept of a second messenger system yet, so let me just very briefly talk about that, just to give you a preview of what we're going to talk about. Up until now, we've seen that the neurotransmitter binds directly to a receptor, and the action of that receptor molecule, such as opening to allow ions flow, causes the response in the postsynaptic cell. In second messenger systems, the effect on the postsynaptic cell is more indirect. So we're still going to have a messenger molecule like the neurotransmitter, like norepinephrine, still going to bind to a receptor molecule. But that receptor in and of itself is not actually going to cause the postsynaptic cell to respond. Instead, the receptor for the neurotransmitter is going to activate a second molecule. And that second molecule, once activated, will then have an effect on the postsynaptic cell. Sometimes there are several layers to this, where the second molecule that's activated then goes on to activate a third molecule. And it's the third molecule that has a direct effect on the cell. And so you have these intermediate or second messengers that carry the message from the neurotransmitter on into the cell and ultimately have the effect. So this is known as a second messenger system. Again, quite common way in which certain kinds of neurotransmitters function. It is also a common way in which hormones have an effect on their target tissues as well. There are a variety of second messenger systems. One of the more common ones involves cyclic AMP, which is a molecule that, of course, is the second messenger in the adrenergic synapse. And cyclic AMP, by the way, AMP stands for adenosine monophosphate. So it's a version of ATP in which we've removed two of the phosphates. So we go from ATP, triphosphate, first to ADP, diphosphate, and then we remove another phosphate and we get AMP, adenosine monophosphate. The cyclic refers to the fact that the phosphate, the single phosphate group, arcs around and part of it binds back to the main portion of the adenosine molecule, forming a cyclic or a cycle type structure. At any rate, that is a second messenger for this particular synapse. So what happens is the norepinephrine diffuses. Again, the release mechanism is the same. The nerve signal triggers exocytosis of synaptic vesicles, releasing norepinephrine. Norepinephrine diffuses across the synaptic cleft and binds to its specific receptor. But in this case, the receptor is not a chemically gated ion channel, but instead it's a transmembrane protein. Remember, transmembrane proteins, also known as intrinsic proteins, are ones that pass completely through the membrane. They don't just sit on the surface, either the outside or inside surface. Those were called extrinsic proteins. Transmembrane proteins are ones that pass all the way through the cell membrane. And this usually, in this particular case, in the adrenergic synapse, the receptor molecule is a transmembrane protein that's complexed with a membrane protein called the G protein. So the G protein is essentially the anchor in the membrane, and the receptor molecule is attached to that G protein. We don't get immediate responses from these types of synapses because it takes a little while for the second messenger to diffuse through the cell and have an effect. So with the previous kind of receptors involving ion channels, of course, those ion channels, those chemically gated ion channels, opened immediately. And so we saw an immediate effect. But in this case, we have to wait a little while for the second messenger to be activated, released, and diffused through the cell to its target site on the cell. So they're a little bit slower than the synapses we're used to thinking about. The second messenger system type mechanisms in general also often involve activation of enzymes within the cell. And by activating multiple enzymes, a single messenger molecule can activate multiple enzymes to perform specific chemical reactions in the postsynaptic cell. We get what's called enzyme amplification. So a single, again, molecule of neurotransmitter can activate multiple enzymes, and then each of those enzymes can catalyze multiple reactions, and so we get this vast amplification of the molecule of the neurotransmitter's effects. So we get a slower response, but often a single neurotransmitter molecule can produce a much more profound response. So here's a diagram of what happens at an adrenergic synapse. Again, nerve impulse travels down to the axon terminal, calcium flows in, activates enzymes, facilitates exocytosis of synaptic vesicles, and the norepinephrine neurotransmitter is released. The norepinephrine will then bind to its receptor molecule, which, notice again, is a transmembrane protein passing through the entire membrane. Here we see the norepinephrine molecule in green binding to its receptor site on the receptor molecule. We said that the receptor is usually associated with a G protein that acts like an anchor, but it's an anchor that can be released. And so what happens is when the norepinephrine binds to the molecule, the G protein is released and migrates, crawls along the inside surface of the cell membrane. So in a way, you can really think of the G protein as being the second messenger in this system. The G protein crawls until it finds another transmembrane protein called adenylate cyclase. Adenylate cyclase is, noticed by the ending ASE, is an enzyme. And so when this G protein crawls and finds an adenylate cyclase, it will bind to it and activate the enzyme, this membranous enzyme. What does this enzyme do? Well, as you might guess, based on prior discussion, this adenylate cyclase enzyme is designed to specifically break ATP down by removing two of its 3-phosphate groups and turns it into cyclic AMP. Now we have cyclic AMP in the cytoplasm of the postsynaptic cell, and that cyclic AMP can do one of several things depending on the specific cell involved. In some cases, the cyclic AMP can migrate to chemically gated channels in the postsynaptic cell, bind to the chemically gated channel from the inside of the cell as opposed to the outside, and again cause a change in shape of that chemically gated channel, which will allow something to flow in. Oftentimes it's an ion channel, and so the cyclic AMP, by binding to the chemically gated ion channel, might, for instance, allow sodium to flow into the cell, and that of course would cause an excitatory postsynaptic potential and might eventually create an action potential if this postsynaptic cell is a neuron. So it can have a similar effect, which is to ultimately trigger an action potential in the postsynaptic neuron. But notice it does so through through a much different route than does direct stimulation of a chemically gated channel by a neurotransmitter. Oftentimes, though, we have other effects of the neurotransmitter through cyclic AMP. A very common mode of action of cyclic AMP, as we hinted at earlier, is that the cyclic AMP may activate specific enzymes in the postsynaptic cell, and this may lead to very specific chemical reactions initiated in the postsynaptic cell, which alter the metabolism or chemical behavior of the postsynaptic cell. It might be, for instance, triggering enzymes that lead to the creation of a hormone. So if this postsynaptic cell, say, is a glandular cell instead of a neuron, this may be how norepinephrine will stimulate that gland to secrete its hormones. And yet a third mode of action for a second messenger like cyclic AMP is that it travels into the nucleus of the postsynaptic cell and triggers genetic transcription. Remember, transcription is the making of an RNA molecule from DNA. And so cyclic AMP can travel into the nucleus and activate enzymes that regulate genetic transcription. And so it may cause the cell to transcribe specific RNA sequences, which then, of course, travel out into the cytoplasm, where we perform translation at a ribosome. Remember, translation is the making of a protein from the information encoded in an RNA strand, a messenger RNA strand of RNA. And so the protein that is synthesized is going to be, of course, a very specific one. Oftentimes, once again, it's a very specific enzyme which will catalyze a very specific chemical reaction in the postsynaptic cell, again altering its metabolism in a very specific way to create some kind of response. So neurotransmitters don't always have to cause an inhibitory or excitatory postsynaptic potential. They don't, in other words, always have to have electrical effects on the postsynaptic cell through these second messenger systems. They can also have metabolic effects on the postsynaptic cells. So that's a more interesting and complicated kind of synapse than the ones we've been looking at. And as I mentioned earlier, hormones also tend to work in this way through second messenger systems, though there are other second messenger systems besides this one involving cyclic AMP. Okay, so we know how we can excite a postsynaptic cell or inhibit it at a synapse. How do we cease the transmission of a signal at a synapse? How do we stop it? And again, whether it's excitatory or inhibitory, eventually we want to stop the signal and have the postsynaptic cell return back to its normal resting state. And this is especially important, of course, for excitatory signals, where we just don't want to continue to excite the postsynaptic cell, which would result in endless firing of neurons, or in the case of skeletal muscle cells, endless contraction of muscle cells. Well, there's a couple of steps involved in stopping the signal, and it's kind of amazing that it can happen so quickly given what has to happen. But it does happen virtually instantaneously. Of course, we stop releasing neurotransmitter from the presynaptic cell. So by simply not sending nerve signals down the axon anymore, we don't have calcium entering the cell, no more activation of the enzymes, and no more stimulation of exocytosis. And so we stop releasing the neurotransmitter. So that's obviously the first thing that must happen. But remember, there's still plenty of neurotransmitter molecules floating around in the synaptic cleft that are capable of continuing to stimulate or bind to the receptors. So how do we deal with that lingering neurotransmitter that's still in the cleft? Well, as you might remember, neurotransmitter, when it binds to its receptor, doesn't bind permanently. It binds for a very short period of time, stimulates the receptor, and then is released. When I say a very short period of time, it's about 1 millisecond. So a neurotransmitter, say acetylcholine, will bind to its acetylcholine receptor, and then is about a millisecond later is released. And then what happens is that neurotransmitter, that acetylcholine, say, will bounce around the synapse some more, and it may bind yet again to a acetylcholine receptor for a millisecond, and then get released again, and then bounce around and bind to another one, and so on and so forth. So The acetylcholine or any neurotransmitter can bounce around inside the synaptic cleft, binding to the receptors multiple times, but again, only for short periods of time, activating them and then releasing. I apologize for the formatting here. This 6, 7, and 8 should just be more bullets. There's really we start with bullets and then go to numbers, and the numbers don't match up, so just ignore that. These are just bullets. All right, so how do we keep this neurotransmitter, which is bouncing around inside the synaptic cleft, how do we eventually get rid of it and keep it from continuing to stimulate the receptors? Well, remember, in many cases, such as in the case of acetylcholine, we have an enzyme known as acetylcholinesterase in the synaptic cleft, which is competing with the acetylcholine receptors for acetylcholine molecules. So this acetylcholine that's bouncing around, just by luck, sometimes it might bond to an acetylcholine receptor, but just by luck, by random fortune, it may sometimes bind to an acetylcholinesterase molecule. Of course, once it binds to the acetylcholinesterase molecule, it is degraded, it is broken down, and so that molecule is no longer available to bind to the receptor molecules. And because the neurotransmitter only binds to the receptors for about a millisecond and then releases again, but binding to the acetylcholinesterase essentially is permanent because it's broken down after that. Very, very quickly, just by chance, many of the acetylcholine molecules are broken down by acetylcholinesterase and are not available anymore to bind to the receptors. After they're degraded, remember, the presynaptic cell will reabsorb the fragments, which is acetate and choline, acetic acid and choline, and reabsorb them and use them, recycle them, and remanufacture acetylcholine. This is very typical for other neurotransmitters as well. Another way of clearing the synaptic cleft of the neurotransmitter is for the axon terminal, the presynaptic cell, to reabsorb the neurotransmitter by endocytosis, to pull it back into the axon terminal and get rid of it that way as well. This doesn't happen in all cases, but in some neurotransmitters this happens. In the case of monoamines, they get reabsorbed, and interestingly enough, they don't get reused. They get broken down after they're reabsorbed, and then the fragments are reassembled, and it seems like a waste of energy and time to reabsorb it in its whole form, break it down, and then rebuild it again. But nevertheless, that's what happens in the case of the monoamine neurotransmitters. Others, like acetylcholine, get reabsorbed in their whole form and simply get repackaged back into synaptic vesicles. And then finally, another way that neurotransmitters can be cleared from the synaptic cleft is for them to simply diffuse out of it. So as they're bouncing around randomly inside the synaptic cleft, inevitably some of them, many of them, bounce around until they literally bounce right out of the cleft into the surrounding tissue space. In the central nervous system, remember, this tissue space is surrounded by astrocytes, which are primarily responsible for maintaining a proper environment for neurons. And so the astrocytes will absorb these neurotransmitter molecules that diffuse out of the cleft and again return them to the neurons so that they're not wasted. This similar job in the peripheral nervous system is performed by the satellite cells, the glial cells that we find in the ganglia of the peripheral nervous system. And so all these different activities help to clear the synaptic cleft of the neurotransmitter very, very quickly, causing an almost immediate, or at least to our senses, almost an immediate ending of the stimulation. And here we see a variety of mechanisms that we talked about. The acetylcholine can get destroyed by acetylcholinesterase. Now, in some synapses, acetylcholinesterase is free-floating in the synapse. In other cases, acetylcholinesterase is bound to the membrane of the postsynaptic cell. And so the neurotransmitter, again, just by chance, either bumps into its receptor molecules, the chemically gated ion channels, or it bumps into an acetylcholine molecule, either free-floating or on the membrane. And of course, once it bumps into that, it's destroyed into, again, acetic acid or acetate and choline. Those breakdown products get reabsorbed and recycled in many cases. Sometimes the acetylcholine or neurotransmitter gets directly reabsorbed back into the terminal by endocytosis. That's not shown here. Sometimes the neurotransmitter diffuses out of the cleft, where it's picked up again by astrocytes or satellite cells and then returned to the neuron. So this only shows a couple of things that can happen. Remember, there's other mechanisms of removal that I just mentioned. All right, at this point we shift focus to neuromodulation. Neuromodulation essentially means the modification of a nerve signal by a chemical. So are these chemicals neurotransmitters? It's hard to say. They're kind of one of those gray areas, one of those in-between kinds of molecules. So neuromodulator is probably the best term we have to distinguish them from true neurotransmitters. Again, they're chemicals secreted by neurons that affect the behavior of neurons but don't function exactly like neurotransmitters. And so by saying affecting other neurons, they can alter or affect them in many ways. They can affect how rapidly those neurons make their own neurotransmitters. They might affect how rapidly those other neurons release their neurotransmitters. They might affect how rapidly the other neurotransmitters are broken down or reabsorbed at the synapse. And so they're basically neuromodulator chemicals adjust the sensitivity and activity of postsynaptic neurons. And you may recall we listed nitric oxide as a neurotransmitter. Earlier, we said a gas, a metabolic gas that can function like a neurotransmitter. Perhaps it would have been better to really talk about it as a neuromodulator, one of these neuromodulators. Again, it's produced normally as a metabolic product of cells, but in this case, in this vicinity, it can enter postsynaptic cells and activate second messenger pathways just like a neurotransmitter would. And this will then make that cell more or less, in this case, more sensitive to the actual neurotransmitter that's going to function like norepinephrine through a second messenger system. So again, by sort of getting the second messenger system going to begin with, it makes that cell more sensitive to, say, a neurotransmitter like norepinephrine when it comes along and activates that same second messenger system. Okay, so some true neurotransmitters that have actual neurotransmitter effect can also act as neuromodulators that, again, slightly inhibit or excite a postsynaptic cell, making it more or less likely then to respond to its actual normal neurotransmitter. So these are called neuromodulators. All right, at this point we are at objective 145, and we're switching focus to the last topic of this chapter, which is neural integration. Neural integration is the ability of neurons to process information, store it, remember it, recall it, and decision making. So if you think back to the beginning, our discussion at the beginning of this chapter, when we talked about the functions of the nervous system, we said there were three functions: the sensation, response, and then in between sensation and response there was integration, association, processing, perception, whatever you like to call it. But basically, how do neurons communicate with each other mostly inside the central nervous system, to do things like think, remember, make decisions, analyze, those kinds of things. This is a ongoing frontier of investigation in anatomy and physiology, and we are still learning quite a bit, of course, about how the brain functions. So this is going to be just scratching the surface of and a very simple presentation of what's involved in neural integration. Okay, so the first thing to remember about interneurons, which do most of the neural integration, is that they're multipolar neurons. They're multipolar neurons, and they can have an incredible number of connections with other interneurons, and this is what allows for complex integration. So remember, some neurons in the brain can have as many as 40,000 contacts with other neurons, and in the cerebellum you can have even more, up to 80,000 contacts with other neurons. So these neurons are incredibly well connected or networked with one another. Now, the one thing that will slow things down is with all these contacts and potentially thousands of synapses involved in a single integrative pathway, there is something known as synaptic delay. You don't really notice it at a single synapse where things seem to be happening virtually instantaneously, but when you have a pathway, an integrated pathway inside the brain, say, that involves 1,000 synapses, the time it takes for each synapse to occur, which is called synaptic delay, adds up, and it could become noticeable. Often doesn't become noticeable, but it does make things travel a little bit more slowly. The most essentially what causes synaptic delay is all the things that have to happen between when the action potential arrives at the axon terminal and the generation of a new action potential in the postsynaptic cell at the trigger zone. So again, between those 2 points, a lot has to happen. Enzymes have to be activated in the axon terminal, exocytosis has to occur, diffusion of the neurotransmitter has to occur, binding to the chemically gated channels has to occur, the channels have to open and ions have to flow, and the sodium that flows in during that graded potential has to spread through the postsynaptic neuron till it arrives at the trigger zone, and then of course that has to cause voltage gated channels to open. So a lot has to happen between the action potential in the presynaptic axon and the new action potential in the postsynaptic axon. And actually it's amazing that synaptic delay isn't longer than it is. All of those events that I just described take place in about a half a millisecond. So again, for a single synapse, it's virtually instantaneous. But if you have 1,000 synapses in a pathway, and that would be admittedly a very big pathway, but theoretically, if you had 1,000 synapses, then if the synaptic delay for each synapse was half a millisecond, the sum total delay would be half a second. So again, not huge. Half a second is still very quick, but it is something that you have to take into consideration. Okay. So neural integration, again, is how we process information and how neurons essentially talk to each other. And it's all based on postsynaptic potentials. It's all based on what's happening in the postsynaptic cells and how those postsynaptic cells receive signals from presynaptic cells and how they respond to it. So remember, for a postsynaptic cell to fire, it has to experience a threshold level of depolarization at its trigger zone. And what initiates that threshold level of depolarization is an excitatory postsynaptic potential coming from the dendrites or cell body of the neuron. And so, as we've described repeatedly throughout this chapter, when you get an excitatory postsynaptic potential, either at dendrites or at the cell body, the sodium that flows in will diffuse through the cell to the trigger zone. And again, if it's powerful enough, if there's enough sodium, it will trigger a new action potential. Also remember that there can be inhibitory postsynaptic potentials as well, caused by, say, chloride entry, which hyperpolarizes it. Now, what we didn't mention is that these two things can be happening at the same time. We've, up to this point, sort of had a very simplistic description where you have a single synapse causing the release of some neurotransmitter molecules at a single site, say between one axon terminal and one dendrite, and then that's spreading towards the axon hillock, the trigger zone. In reality, you usually have multiple axon terminals involved in synaptic transmission in multiple places on the postsynaptic neuron. So many dendrites involved in active synapses with many axon terminals all at the same time. And what we haven't said is some of those synapses might be generating excitatory postsynaptic potentials. Others of those synapses might be causing inhibitory postsynaptic potentials all at the same time. And so what ultimately is going to arrive at the trigger zone of that single postsynaptic cell is some combination of all the different EPSPs and IPSPs that might be occurring at its dendrites and cell bodies simultaneously. And this really gets now to the heart of integration, neural integration. So it's not a simple situation as we might have been hinting at, where you get some neurotransmitter released at a single synapse, and that's going to cause a postsynaptic cell to fire. It's just not that simple. It's the sum effect of multiple synapses, some, again, excitatory, some inhibitory. It's the combination of all those effects that determines what ultimately ends up at the trigger zone and whether that postsynaptic cell fires or doesn't fire. Right, and so here again we see the two types of postsynaptic potentials that we can get. Remember, an EPSP, an excitatory postsynaptic potential, depolarizes the membrane to some degree. They're graded potentials so that they can exist at any size. An inhibitory postsynaptic potential caused, for instance, by the opening of chloride channels causes a hyperpolarization, as we've already described. And again, these are graded as well and can exist in any size. All right, and again, as we've said, different neurotransmitters can cause different kinds of reactions, and as we said, even the same neurotransmitter can cause excitation in some cells and inhibition in others. Right, so acetylcholine, for instance, we know excites skeletal muscle cells, but interestingly enough, when it is released onto cardiac muscle tissue, it inhibits cardiac muscle tissue. And so again, this is all review, things we've said before. Different neurotransmitters can have different effects on different cells. The same neurotransmitter can have different effects on different cells. And again, it really ultimately depends on what kind of receptors you have for those neurotransmitters. Now, we talked about the concept of summation when we talked about muscle physiology. Remember, summation is how we, in muscles, is how we combine muscle twitches into a whole muscle response. And we use that to affect the force of contraction or the duration of muscle contraction. Well, we can also experience summation in neurons. And with the concept of summation with regard to neurons is how we add or combine graded potentials to have an effect on the postsynaptic potential. So remember, in the previous couple of slides ago, I said that it's the combination of all the different excitatory and inhibitory postsynaptic potentials that's going to determine what actually happens at the trigger zone. Well, that is essentially what we mean by summation, the adding up or combination of the excitatory and inhibitory, all the different excitatory and inhibitory postsynaptic potentials to produce a single net effect at the trigger zone. All right, and so will a neuron fire if it receives a bunch of excitatory signals from neurotransmitter molecules, produce a lot of excitatory postsynaptic potentials? Maybe. It may be that those sum up enough to create a threshold depolarization at the trigger zone, or maybe not. If that's happening at the same time that we're getting a lot of inhibitory postsynaptic potentials, the net effect of the two might be to cause actually no change at the trigger zone, and we see nothing happen in the postsynaptic cell. So summation simply means how are we going to combine these excitatory and inhibitory postsynaptic potentials to create an effect or no effect on the postsynaptic cell. With temporal summation, and again, remember we saw both temporal and spatial summation with muscles have similar phenomena in the neurons. Temporal summation occurs when you get multiple EPSPs from a single synapse because of a high frequency of stimulation. Remember, in muscles, temporal summation led to tetanic contraction due to high frequency of stimulation. Temporal summation in neurons means that the postsynaptic cell is going to receive a high frequency of stimulation from the presynaptic cell. Okay, so this is not adding up EPSPs from multiple synapses. Temporal summation occurs at a single synapse where we just stimulate that synapse over and over and over again, 10, 20, 30 times a second. Excuse me. And so you're starting to get excitatory postsynaptic potentials generated so quickly that new ones begin before the previous ones even end. This allows their effects to add up to create a much larger effect that then can spread through the cell and trigger a threshold level of depolarization at the trigger zone. And so it makes it much more likely to get an action potential when we have temporal summation of the EPSPs. By the way, you can also get temporal summation of IPSPs, inhibitory potentials, to make it much less likely that the postsynaptic cell will fire. All right, spatial summation, of course, involves multiple synapses. All right, so it's not a high frequency of stimulation, it's just a high magnitude of stimulation. And so we're going to get many EPSPs occurring at many different synapses on the postsynaptic cell. So in other words, the postsynaptic cell is receiving EPSPs from multiple synapses, sometimes hundreds, potentially thousands of synapses all at the same time. And so you're going to get a flood of sodium into that postsynaptic cell because of the sheer number of synapses that are occurring simultaneously in the case of excitatory postsynaptic potentials. And all that sodium is going to collectively migrate or diffuse towards the trigger zone. And this massive amount of sodium that's been introduced to the cell is certainly going to be enough to reach threshold at the trigger zone and cause firing. And once again, you can get spatial summation of inhibitory postsynaptic potentials as well, and this can make the postsynaptic cell much less likely to fire. So you can get summation both for excitation and inhibition as well. Here we see examples of temporal and spatial summation. Again, in temporal summation, you've got a single postsynaptic cell here, and here are several presynaptic cells that are synapsing simultaneously on the postsynaptic neuron. In remember, in temporal summation, you're just looking at the effects at a single synapse, this one right here, and we get this high frequency of let's say excitatory postsynaptic potentials hitting the postsynaptic cell in one spot, one synapse. But they happen so frequently, they just continue to cause sodium to flood into the cell, flood in. That sodium then spreads to the trigger zone and creates then a high frequency of action potentials in the postsynaptic cell. In spatial summation, we don't have a particularly high frequency of stimulation, but we have multiple presynaptic axons firing at the same time, causing sodium to enter in multiple synapses. Now, the amount of sodium that's entering in each synapse is not as great as it was for this one synapse, but if you collectively add up the effects of all these different synapses, especially if there's hundreds of them, again, that can result in a flood of sodium into the cell, which then by the time it diffuses to the trigger zone will certainly be enough to trigger a new action potential. And again, we see a high frequency of action potentials. So these are the two ways that we can integrate signals, combine signals, process signals to create postsynaptic potentials from presynaptic neurons. Here we see a graphic representation of how excitatory postsynaptic potentials might sum. Again, this could be temporal or spatial summation. We just see how multiple excitatory postsynaptic potentials, be they coming very rapidly at a single synapse or coming from different synapses, how they will add up and eventually make it much easier to achieve threshold depolarization in the postsynaptic cell. All right, you've seen perhaps a couple of times throughout these PowerPoints the idea of facilitation. What is facilitation? Well, presynaptic facilitation, or facilitation in general, means making it much more likely that a postsynaptic cell will fire. Essentially, that's what facilitation is making a postsynaptic cell much more likely to fire. Presynaptic facilitation simply means that a presynaptic neuron is going to enhance the ability of another neuron to make a postsynaptic cell fire. So in other words, you've got a synapse between a presynaptic cell and a postsynaptic cell, and you've got another neuron, a third neuron, that can do something to increase the likelihood that the presynaptic neuron will excite the postsynaptic neuron. This is what we call presynaptic facilitation, increase in the likelihood that a presynaptic cell can stimulate a postsynaptic cell. The third neuron in this case that can do this is called the facilitating neuron. The facilitating neuron. And there are a couple of ways in which this can happen. A common way is through the release of the chemical serotonin. So notice in this diagram, we're showing one common kind of presynaptic facilitation. Here we have a typical presynaptic cell with its axon terminal. This represents the postsynaptic membrane. Here's the synaptic cleft between the two. So you've got a atypical synapse, and here is the voltage-gated calcium channel. Remember that has to let calcium in to trigger exocytosis. Notice here we have a third neuron, the facilitating neuron, that is essentially synapsing or communicating with the presynaptic axon terminal. And so what happens is, if I have a signal coming down this facilitating neuron, it is going to release neurotransmitters onto the axon terminal. And here's something that, again, we're just not used to seeing. This is new for us. And the axon terminal can actually be stimulated by these neurotransmitter molecules. Now, in the case of serotonin, let's say this is serotonin, what serotonin will do is it will diffuse into the cell and cause these calcium channels to remain open longer than they normally would. And by causing the calcium channels to stay open longer than they normally would, we get a greater degree of exocytosis and neurotransmitter released at the synapse than you normally would from a single nerve signal. And so what this facilitating neuron is actually doing is changing the behavior of this presynaptic axon terminal, causing it to release more neurotransmitter than it normally would. Well, by releasing more neurotransmitter, it's going to have a greater effect on the postsynaptic cell membrane. And so we have facilitated this synapse, right? This facilitating neuron has caused changes in the axon terminal, which has facilitated this synapse. In other words, made it more likely for this presynaptic cell to stimulate the postsynaptic cell. This is another aspect of neural integration. Again, how we regulate and control how interneurons especially communicate with one another. The opposite of presynaptic facilitation, of course, would be presynaptic inhibition. In presynaptic inhibition, we have the same basic elements, but instead of the third neuron releasing a chemical that enhances synaptic transmission, instead the third neuron releases a chemical that tends to inhibit synaptic transmission between the pre and postsynaptic fibers. And so in this case, it might prevent or quicken, or shorten, I should say, how long the calcium channels stay open, thereby causing less neurotransmitter to be released. And so diagrammatically, what we see is a diagram very similar to the previous one. So this diagram looks almost identical to the previous one, except this third neuron now is not called a facilitating neuron, but is called an inhibitory neuron. And it might serve to, again, dampen or reduce synaptic transmission. And so in this case, what we might have is we've got an active synapse and the calcium channels open, and calcium's coming in and stimulating exocytosis, typical synapse. But when this inhibitory neuron fires, it might release a neurotransmitter that causes an inhibitory postsynaptic potential in the axon terminal. This inhibitory postsynaptic potential then essentially counterbalances the excitatory signal coming down. They neutralize each other. The calcium channels then close, because remember, these are voltage-gated channels. And the IPSP, by neutralizing the excitatory signal coming down, essentially balancing it out, now there's no voltage change to cause the calcium channel to open. So even though this axon terminal is receiving a nerve signal, it's having no effect, because that nerve signal is canceled out by the effect of this inhibiting neuron. And so the calcium channels don't open, and we do not get exocytosis at this target synapse. And so now a synapse that would normally fire in response to a nerve signal is not going to fire. And so that's inhibition of that synapse by a third neuron. This is known as presynaptic inhibition. And again, you've got all kinds of possibilities with simultaneous inhibition and facilitation, and the net effect of all this determines whether a particular synapse is functional or not. And these are all aspects of neural integration and how we control the spread of neural signals from one interneuron to another during higher processing within the brain. Neural coding, very simply, is the topic of how the brain converts information into a meaningful pattern of action potentials. So we understand now that through facilitation and inhibition and summation, we regulate how action potentials travel between one neuron and another. But how do we make meaning out of those action potentials? How do we put some kind of sensibility or meaning to what all those action potentials are supposed to indicate to us? Well, there's two kinds of information that can, two ways in which information can be coded in other words, two ways in which we can make sense of these patterns of action potentials. Qualitative information is a coding that's based on which neurons fire. Of all the neurons that are in the brain, which ones are actually firing? This is going to tell us what kind of information we're getting. Not so much how strong the signal is, but what kind of information is it? Right? So if I'm receiving action potentials, for instance, from a sensory neuron that's in the retina, I know that those action potentials coming from those neurons should be interpreted as light. If they're coming from sensory neurons like, say, Pacinian corpuscles in the skin, I know that those action potentials should be interpreted as pressure. So information is coded based on which neurons are firing. And we have somehow some ability to understand that action potentials in different neurons mean different kinds of information. In terms of signal strength, that's known as quantitative information. And so the information about the strength or intensity of a signal can be coded in two different ways. One is the frequency of the stimulation. Generally speaking, lower frequencies of action potentials mean weaker signals, weaker stimuli. Higher frequencies of action potentials means a greater strength of signal. And so we interpret frequency of action potentials as a way of understanding strength of signal. The other way we interpret it, again, is based on different types of neurons. We actually have in our brains neurons with different thresholds. Now, previously I've said the threshold depolarization is usually about 15 millivolts less than resting. But that's not always the case. We have some neurons with higher thresholds that are more difficult to stimulate, to trigger. Well, of course, if we start stimulating those neurons with the higher thresholds, that must mean that we're experiencing a stronger signal. And so just by virtue of activating some of these higher threshold neurons, that also is understood by the brain to mean a stronger signal. So the type of neuron can help us understand the qualitative type of information we're receiving and the strength of the signal. And frequency of action potentials helps us understand the strength of the signal as well. And here we see a frequency of action potentials coding for strength of signal. All right, we're going to finish up by talking about neural pools and memory. A neural pool, and we're going to have to move quickly through this section since we're almost out of time. A neural pool is essentially a group of neurons that act together, a group of functionally related neurons. And again, it can be several hundred or it can be several thousand, and they're usually associated with a particular function. So nuclei, remember we had nuclei in the brain. We talked about, and you will talk about this more in A&P2, we have nuclei that control heart rate or nuclei that control respiratory rate. These are neuronal pools or neural pools of neurons working together to control a specific function. Now, neurons can have what are called discharge zones and facilitated zones. A discharge zone is anything that a neuron can by itself activate. So a neuron's discharge zone is whatever it can activate all by itself. So a discharge zone may be a single postsynaptic cell, it may be a few postsynaptic cells, it may be several hundred postsynaptic cells. It is possible for a single neuron to have an axon that branches multiple times, kind of like a motor unit, if you think about it, and stimulate hundreds to thousands of postsynaptic neurons. But whatever the neuron can activate all by itself, that is known as the discharge zone of the neuron. On the other hand, some neurons also have what are called facilitated zones. Facilitated zones are what the neuron, synapses that the neuron forms, that it can influence but not actually trigger all by itself. So in the neuron's facilitated zone, the neuron can contribute to creating postsynaptic triggering, but it has to work with other input neurons to trigger the postsynaptic cells. And so a single neuron usually has a more narrow discharge zone, relatively fewer neurons that can actually stimulate all by itself, and then it tends to have a broader or wider facilitated zone, many more neurons that it can stimulate, working only with the help of other neurons. And so here we see a graphical representation of this, a single neuron with a branching axon. The postsynaptic cells that it can trigger all by itself are within that neuron's discharge zone. Other neurons here that it can't quite trigger all by itself, but it needs help of other neurons sending signals in. That's known as the facilitated zone of the neuron, and notice that's usually broader than the discharge zone. And so using these discharge zones and facilitated zones, we can have different types of neural pathways or neural circuits. And again, these are like electrical paths that action potentials can follow. And you see here there's four basic kinds of neural circuits. Diverging circuits, of course, is where one axon branches and synapses with several postsynaptic cells, right, as we were just talking about, discharge zones and facilitated zones. And as we said, one neuron can have potentially output through hundreds of postsynaptic neurons, maybe even thousands in some cases. A converging circuit is where many different axons from different input neurons synapse on a single postsynaptic cell. So this is the opposite of a diverging circuit. Here, many presynaptic fibers converge on a single postsynaptic cell. And this is, of course, how we get facilitated zones potentially where multiple neurons work together to stimulate a postsynaptic cell. Those are the two simple kinds. Then we have reverberating circuits. In reverberating circuits, you get sort of a linear sequence of action potentials where one neuron triggers a second, which then triggers a third, and a fourth, and a fifth in a linear fashion. But at some point, some of the downstream axons, some of the downstream cells will cycle back and re-stimulate some of the earlier cells in the sequence. And so the signal sort of does a loop and comes back and triggers itself again. And it keeps triggering itself over and over and over again to create this cycle of excitation that seemingly never ends. This is a very useful kind of circuit when you have repetitive cyclic kinds of activities. So we see reverberating circuits, for instance, in the diaphragm and intercostal muscles. These are muscles involved in the repetitive action of breathing. And so you don't have to think about it so much. The circuit itself is designed to reverberate or cycle by having later neurons in the sequence re-stimulate earlier neurons in the sequence. So a self-cycling kind of circuit. And then finally, our fourth kind is a parallel after discharge circuit, a big name. Basically what happens is a single input neuron diverges, like in a diverging circuit, to stimulate several postsynaptic cells. So very much like a diverging circuit to begin with, but instead of just stimulating a single postsynaptic cell, it stimulates a postsynaptic cell that leads to a chain of stimulation. And so each chain has a number of different synapses that it can lead to, and eventually these all reconverge onto one or a few output neurons with varying degrees of delay. So that one is a hard one to visualize. We have to see a diagram of it. And so here we see our four types of circuits. The diverging circuit, where a single input neuron spreads its signal out to multiple postsynaptic cells. The converging circuit, where you have multiple input neurons converging on a single postsynaptic target. The reverberating circuit, where you have a chain of synapses, and somewhere in that chain we get a branch that comes back and re-stimulates the early neuron to keep the cycle going. And then the parallel after discharge, essentially you start with a diverging circuit, which leads to a chain of varying lengths, chains of synapses, which then converge back on a single postsynaptic cell. So really the best way to describe the parallel after discharge is a diverging circuit followed by a converging circuit. Okay, in terms of how we process action potentials through these circuits, we can have serial processing or parallel processing. In serial processing, essentially, again, it's fairly linear, and so you have action potentials carried along a pathway in a linear fashion. And so this is the kind of processing we do when you can only process the flow of a single kind of information at a time. Your book uses the idea of reading a book. You can't read a book and do much else at the same time. You just have to focus on reading a book. So this is sort of a way of what we think about focused thinking. So when you're doing math, when you're trying to solve, say, an algebra problem, it's not like you can multitask and do a lot of other things while you're doing that. It requires your focus, and so we're doing serial processing. Now we can jump back and forth. You can look up from your algebra problem and realize something's on fire on the stove and change your focus. But while you're doing the problem itself, you can't really be putting out the fire at the same time. All right, that's serial processing. Parallel processing is done more through things like diverging circuits and parallel circuits that we talked about, where you've got information traveling along multiple circuits simultaneously, multiple pathways simultaneously. And so this kind of processing is what we do when we multitask, when we can do many things at the same time. So for instance, it's not uncommon for people to be able to type and have a conversation at the same time. It's not easy, but it's theoretically possible. Some people who are good at typing can do it. Singing and playing a musical instrument at the same time. Again, not easy, but many people obviously can sing and play an instrument at the same time. Two very different things that are happening, but they're happening simultaneously. So this is what we use parallel processing for. We want to do multiple tasks at the same time. And again, this is what we generally refer to as multitasking. And different people seem to be better at one of these than the other. So you have the stereotype of the absent-minded professor who is so good at serial processing, so focused on one thing that he or she may not notice other things that are going on around them. And then we have the people that are really good at multitasking but don't seem to be able to remain focused very well on a single item at a time. And again, so there may be actual differences in brain structure in terms of the kinds of circuits we have that allow serial processing and parallel processing. Our last topic is memory. A memory basically is just a specific path that action potentials take through a specific sequence of interneurons. This is called a memory trace or an engram. And of course, these pathways have to be reinforced in order for them to be activated. And so memorization, the process of actually remembering something, involves synaptic potentiation, making it much easier for those synapses to fire, so we don't have to work so hard to activate that pathway, that memory. And of course, these pathways are plastic as our memories are modified over the course of time. In terms of the kinds of memory we have immediate, short, and long-term memory, three different types of memory associated with three different levels of potentiation. Immediate memory is your ability to hold something in your thoughts for a few seconds only. And this is involved in things like reading, just recognizing a word, and understanding its meaning almost instantaneously as you read it. And these very quick immediate memories are probably based on reverberating circuits that reinforce themselves for just a few seconds in the mind. Of course, to be a functional human being, we have to have more than just immediate memory. We have to be able to recall things for more than just a few seconds. So the next level is short-term memory, which lasts from a few seconds to a few hours. This is most of what our working memory is when we're actually performing a task, like calling a phone number you just looked up, or remembering what pi is when you're trying to work through, say, calculate the area of a circle. Well, it's pi r squared. Oh, what's pi? 3.14. You may not remember that. You may have had to look that up, but then as you go through the problem, you have to continue to remember that pi is 3.14. So that's short-term memory. We think short-term memory is the result of titanic stimulation of facilitated synapses. So we use facilitating neurons to create the titanic stimulation, causing calcium to accumulate in the axon terminal, which leads to lots of firing, of course, and lots of neurotransmitter released. Then, of course, to complete the picture, we need long-term memory. Long-term memory lasts for days, months, potentially even a lifetime. And there's different types of long-term memory. Explicit long-term memory is something that we can put words to, essentially, that you actually can focus on and think about and recall. Implicit long-term memory is, I guess, a kind of unconscious memory where you don't necessarily have to put words to it. It's just sort of in there. So, for instance, if I want to hammer a nail into a board, I don't have to stop and think about how to hammer. That motor skill is built into my long-term memory, and once I've learned how to do it, I just do it, and I don't even think about it, and I don't have to stop to remember how to do it. Emotional memories are also this kind of implicit long-term memory. Again, you don't necessarily have words for it, you just know you're afraid or excited or happy or whatever the feeling is. Long-term memory involves a couple of things as far as we know. Again, we're learning about this as we go. So we know for certain that we have to remodel synapses and create new branching pathways to etch an engram or a long-term memory in place. Other times it involves molecular changes, especially at GABAergic synapse. And so long-term potentiation involves GABA receptors, specifically on dendrites of pyramidal cells, a special kind of neuron in the central nervous system, which we didn't have to talk about. But at any rate, they appear to bind glutamate, and this again alters calcium levels in the axon terminal, and it also can alter calcium levels in the postsynaptic cell. Then the calcium acts as a second messenger system, which affects the postsynaptic cell and through various means facilitating it. Again, we're learning a lot about memory still, and so we're just really scratching the surface here in this discussion. Okay, so that brings us to the end of chapter 12 in our books, and completes our initial discussion of the nervous system. I don't believe that there will be any other videos for this semester because we're at the end of our time together. So it's been a pleasure, and I will communicate with you via email for instructions regarding the final.